Organisms can be made up of large organic compounds that can be grouped into four major classes: carbohydrates, proteins, nucleic acids, and lipids. Of these four classes, only three are further classified as macromolecules. Carbohydrates, proteins, and nucleic acids are referred to as macromolecules because they are each assembled using a unique set of building blocks called monomers. Multiple monomers are linked together with covalent bonds to form polymers. Let's discuss two general reactions. A dehydration reaction is used to synthesize polymers. Consider a short polymer and a monomer. To attach the monomer onto the polymer donates hydrogen and the polymer donates a hydroxyl group. This results in the release of a water molecule and the formation of a covalent bond that links the monomer to the polymer. The end result is the synthesis of a larger polymer. A chemical reaction called hydrolysis is used to break down polymers. Suppose we have a polymer which we would like to break down. When water is added to a covalent bond, one of the hydrogen atoms is attached onto the monomer, and the remaining hydroxyl group is attached onto the polymer. This results in the separation of a monomer from a polymer. Keep in mind that the dehydration reaction and hydrolysis are reverse reactions. Now we will move on to discuss the details of three macromolecules, then complete this lecture with a discussion of lipids. Let's start with carbohydrates. Carbohydrates are organic molecules that provide energy and structural support. Carbohydrates are composed of three main elements: carbon, hydrogen, and oxygen. Almost always, the ratio of elements is one carbon atom to two hydrogen atoms to one oxygen atom. In other words, the formula for carbohydrates is always in multiples of CH2O. For example, glucose is a carbohydrate with formula C6H12O6. Notice that the ratio of atoms is 1 to 2 to 1. Carbohydrates are classified as macromolecules. For this reason, carbohydrates have a unique set of monomers referred to as monosaccharides. Two monosaccharides can undergo the dehydration reaction to form a disaccharide. An example is when two monosaccharides, glucose, fructose, combine via the dehydration reaction to form a disaccharide, sucrose, and water. The bond that links any two monosaccharide together is known as a glycosidic linkage. The reverse reaction is also possible. Sucrose can be broken down into glucose and fructose by hydrolysis. If three or more monosaccharides are linked together by dehydration reaction, the final molecule is called a polysaccharide. Polysaccharides are categorized according to function. They are either used for energy storage or structural purposes. When organisms have excess glucose, they store it as energy storage polysaccharides. Two energy storage polysaccharides include starch, found in plants, and glycogen, found in the muscle and liver of animals. Keep in mind that although starch and glycogen are found in different organisms, they are both composed of glucose monosaccharides held together by glycosidic linkages. Organisms use structural polysaccharides for structural support and protection. The two structural polysaccharides include cellulose, found in cell wall of plants, and chitin found in the exoskeleton of arthropods. Cellulose is composed of glucose monomers, and chitin is composed of N-acetylglucosamine monomers. Let's move on to our next class of macromolecules. Proteins are organic molecules with a wide range of functions. They speed up chemical reactions, provide structural support, transport substances, facilitate cell signaling, and much more. As we go through the course, we will encounter multiple examples of proteins with these functions. You do not need to memorize these functions. Proteins are composed of 5 main elements: carbon, hydrogen, oxygen, nitrogen, and sulfur. These elements do not have to be in specific ratios. Before we discuss further details of proteins, let's define a term. Catalysts are substances that speed up chemical reactions, but are not consumed in the process. There are many types of catalysts. We will represent all catalysts with a large circle. In biology, we are interested in a subcategory of catalysts referred to as enzymes, represented here by a smaller circle within the large circle. Enzymes are proteins that speed up chemical reactions without being consumed in the process. An example of an enzyme is sucrase. Enzymes usually have the suffix ase. Let's see how this enzyme speeds up hydrolysis. The first step is the binding of sucrose onto a specific region of the enzyme referred to as the active site. The sucrose is referred to as the substrate. Next, water enters the active site. The enzyme speeds up the rate at which water is added to the glycosidic linkage. Ultimately, sucrose is broken down into 2 monomers, glucose and fructose. Notice sucrase speeds up hydrolysis and is not consumed in the process. It is ready to take in another sucrose and break its glycosidic linkage. Now let's move on to talk about the details of a protein. The monomer of a protein is called an amino acid. The structure is represented here. The central carbon is bound to 4 different constituents: a hydrogen atom, amine group, carboxyl group, and R group. There are 20 different amino acids, each with a different R group attached to the central carbon. Amino acids can be categorized as either hydrophilic or hydrophobic. Hydrophilic amino acids are attracted to water because their R groups are either polar or charged. Hydrophobic amino acids are not attracted to water because their R groups are nonpolar. Shown here are examples of selected amino acids. You do not need to memorize the name or structure of amino acids for this course. When 2 amino acids combine via dehydration reaction, a dipeptide and water molecule result. The covalent bond that joins the amino acids is known as a peptide bond. If 3 or more amino acids are linked together by dehydration reactions, the final molecule is called a polypeptide. A protein consists of 1 or more polypeptides. The 3-dimensional shape of a protein determines its function. For instance, the enzyme sucrase needs to have a particular 3-dimensional structure to be able to accommodate sucrose in its active site. With the correct structure, sucrase is able to catalyze the hydrolysis of sucrose. Because structure dictates function in proteins, it is important to examine the 4 main contributors of protein structure. Biologists have categorized these contributors into a 4-tiered system. The primary structure is the sequence of amino acids in a polypeptide. The sequence of amino acids is particularly important, and a minor change in sequence can negatively impact the structure and function of a protein. Shown here is an example of a primary structure of a polypeptide. Keep in mind, the covalent bonds that link amino acids together are referred to as peptide bonds. The secondary structure is a local three-dimensional conformation within a single polypeptide. Conformations that make up the secondary structure include alpha helices and beta pleated sheets. In the alpha helix, notice the oxygen in the carboxyl group forms a hydrogen bond with the hydrogen of the amino group. The same interaction is also present in the beta pleated sheets. In summary, these structures result from the hydrogen bonds between oxygen of the carboxyl group and hydrogen of the amino group. Hydrogen bonds that make up the secondary structure exist within a single polypeptide, and the R groups are not involved in the secondary structure. The tertiary structure is the overall three-dimensional conformation of a single polypeptide. 4 different types of R group interactions make the tertiary structure possible. These interactions include hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bonds. Keep in mind these interactions exist within a single polypeptide, and the resulting three-dimensional structure determines the function of the protein. The quaternary structure is the overall three-dimensional conformation of a protein with 2 or more polypeptides. This structure results from the same 4 R group interactions found in the tertiary structure. The only difference is that these interactions take place between 2 or more polypeptides. The structure of a protein is particularly important. Represented here is a correctly folded protein. When there is an increase in temperature or change in pH, the protein can lose its structure in a process called denaturation. The denatured protein does not function correctly. If the temperature and pH are returned to normal, the protein can fold correctly into its original conformation in a process called renaturation. This process can be sped up with a protein called chaperonin. Let's see how the chaperonin can assist in the folding process. Represented here is the chaperonin. It consists of a cap and hollow cylinder. The process starts when an unfolded or incorrectly folded protein enters the hollow cylinder. The cap closes the cylinder to create an interior environment that expedites the folding process. Once the protein is correctly folded, the cap detaches and the correctly folded protein exits. Now let's discuss how a minor change in the primary structure of a protein can affect its structure and function. We will use sickle cell disease as an example. The normal hemoglobin protein has a primary structure shown here. The sickle cell hemoglobin, on the other hand, has a substitution of the amino acid glutamate, GLU, with valine, VAL, at position 6. This switch in amino acid has a tremendous impact on protein structure because glutamate is a polar amino acid whereas valine is a nonpolar amino acid. Let's first examine the normal hemoglobin in further detail. Because glutamate is polar, the final structure of hemoglobin is a distinct unit that can interact with surrounding water molecules. Distinct hemoglobin molecules can carry oxygen effectively. Red blood cells carrying normal hemoglobin have a biconcave disc shape. When the polar glutamate is substituted with a nonpolar valine, the resulting structure is referred to as a sickle cell hemoglobin. These mutated hemoglobins clump together to form a rod-shaped structure because the nonpolar valine cannot interact with the surrounding water. Sickle cell hemoglobins do not carry oxygen effectively. Red blood cells carrying these rod-shaped structures are sickle-shaped. Individuals with sickle cell disease suffer from inadequate oxygen delivery to their tissues because sickle cell hemoglobins are ineffective oxygen carriers. We will now move on to our last macromolecule. Nucleic acids are organic molecules that store and transmit genetic information. They are composed of carbon, oxygen, hydrogen, nitrogen, and phosphorus, in no specific ratio. The monomer of a nucleic acid is called a nucleotide. The structure of a nucleotide is shown here. A 5-carbon sugar is attached to a phosphate group and nitrogenous base. There are 2 types of nucleotides, ribonucleotide and deoxyribonucleotide. They are differentiated by their 5-carbon sugar and nitrogenous bases. For ribonucleotide, the 5-carbon sugar is called a ribose, and the possible nitrogenous bases are adenine, guanine, uracil, and cytosine. Adenine and guanine have double ring structures and are referred to as purines. Uracil and cytosine have single ring structures and are referred to as pyrimidines. For deoxyribonucleotide, the 5-carbon sugar is called deoxyribose, and the possible nitrogenous bases are adenine, guanine, thymine, and cytosine. Again, adenine and guanine have double ring structures and are referred to as purines. Thymine and cytosine have single ring structures and are referred to as pyrimidines. Ribonucleotides can be linked by dehydration reactions to form a polynucleotide. Polynucleotides composed of ribonucleotide monomers are called ribonucleic acids, or RNA for short. Notice this RNA molecule has directionality. There is a 5' end with a phosphate group and a 3' end with a hydroxyl group. Although the RNA represented here is single-stranded, RNA can form more complex double-stranded structures. It is out of the scope of this course to discuss complex RNA structures. Deoxyribonucleotides can also be linked to form a polynucleotide. Polynucleotides are composed of deoxyribonucleotides and are referred to as deoxyribonucleic acids, or DNA for short. DNA is double-stranded and forms a helical structure. Adenine always pairs with thymine, and they are linked by 2 hydrogen bonds. Guanine always pairs with cytosine, and they are linked by 3 hydrogen bonds. Notice the DNA strands also have directionality. One strand has a 5' end with a phosphate group at the top and 3' end with a hydroxyl group at the bottom. The other strand is reversed. The 3' end is at the top and the 5' end is at the bottom. We say that DNA strands are arranged in an anti-parallel fashion. The last category of organic molecules is the lipid. Lipids are organic molecules that store energy and facilitate cell signaling. All lipids share the same characteristic in that they do not dissolve in water. In other words, they are hydrophobic. They are composed of carbon, hydrogen, and oxygen. Let's move on to discuss our first category of lipids, fats. Fats are the most effective energy storage organic molecules. 1 gram of fat can store a considerably larger amount of energy than 1 gram of protein or carbohydrate. A fat is composed of 1 glycerol attached to 3 fatty acids. The bond that connects the glycerol and a fatty acid is called the ester linkage. Notice the fatty acids shown here only contain single bonds between the carbon atoms. These fatty acids are called saturated fatty acids and are straight-chained. Because the straight chains pack together tightly, fats containing saturated fatty acids are solid at room temperature. An example is animal fat. Represented on the right is a fat containing fatty acids with double bonds. These fatty acids are called unsaturated fatty acids and are kinked. Because kinked chains cannot pack together tightly, fats with unsaturated fatty acids are liquid at room temperature. An example is vegetable oil. Steroids are the next category of lipids that we will discuss. Steroids are involved in cell signaling and control plasma membrane fluidity. We will discuss these functions in further detail in later sections and focus first on the structure of steroids. All steroids have the same basic structure: 4 fused carbon rings. Shown here is the structure of cholesterol. Cholesterol is a steroid that serves as a precursor for all other steroids, including testosterone, estradiol, aldosterone. Phospholipids are the last category of lipids. These organic molecules are the main components of the cell membrane. Let's examine the structure of a single phospholipid. They are composed of 1 glycerol molecule connected to 2 fatty acids and 1 phosphate group. Phospholipids are a unique class of lipids because they are amphipathic, which means they have both a hydrophobic and hydrophilic region. The tail end is hydrophobic because fatty acids are nonpolar, and the head is hydrophilic because of the negatively charged phosphate group. In the next section, we will see how the amphipathic structure of a phospholipid makes it an ideal molecule for the cell membrane.